Syngeneic tumor models are important preclinical research tools for immuno-oncology drug discovery. However, challenges may arise that can prevent researchers from conducting successful syngeneic studies. In this video, we will review the generation of syngeneic tumor models, the potential issues arising from immunogenicity in syngeneic tumor studies, and how Taconix's JH mouse can enable researchers to conduct these studies without the negative effects of immunogenicity. To create a syngeneic tumor model, a murine tumor cell line that originated in an inbred strain is first cultured in vitro and then implanted into mice of the same genetic background. A key advantage of this type of tumor model is that the host has a competent immune system, which is required to evaluate immune-modulating drugs. One challenge that researchers may encounter when studying novel therapeutics in syngeneic tumor models is immunogenicity. As it relates to drug discovery and development, immunogenicity is the development of an immune response to a biotherapeutic. Biologics can induce both cellular and humoral immune responses in the host. As part of the humoral immune response, the host can develop anti-drug antibodies, or ADA, which can cause various problems. Anti-drug antibodies can reduce the efficacy of a biologic drug in several ways. One example is by decreasing the half-life of the drug. Neutralizing anti-drug antibodies bind an epitope close to the drug site of action, directly blocking the biological activity of the drug. In some cases, this ADA reaction can even cause anaphylaxis. ADA is not only a problem in the clinic, but can also impact preclinical drug discovery. Many immuno-oncology therapies are antibodies or derived from antibody fragments. These antibodies are often humanized or fully human antibodies. Although species differences in epitopes can limit use of these drugs in preclinical mouse systems, there may be sufficient cross-reactivity with the mouse epitopes to permit testing of these drugs in syngeneic tumor models. Some types of immuno-oncology therapeutics may be at higher risk of provoking ADA reactions in the mouse host, and this humoral immune response can mask efficacy of the drug. Repeated dosing as per a clinically relevant dosing schedule may even trigger anaphylaxis in mice. ADA can therefore prevent researchers from determining the efficacy of a biotherapeutic. One solution to this problem is to perform the syngeneic tumor study in a mouse strain that lacks B cells and cannot produce antibodies. Taconix's JH mouse lacks B cells but retains other immune cell types such as T cells, which are important for evaluation of immunotherapies. For biologic drugs which are immunogenic in wild-type mice, the JH mouse provides a system in which to evaluate efficacy without the interference of anti-drug antibodies. Because the JH mouse cannot produce anti-drug antibodies, pharmacokinetics and biological activity of the test article are not affected. This model can also accommodate repeat dosing without the risk of antibody-mediated anaphylaxis. Compared to standard inbred strains, the JH mouse offers a simple solution to interference by ADA in syngeneic tumor studies and permits evaluation of the true efficacy of a biotherapeutic. The JH mouse is available on two inbred strain backgrounds, BALB/c and C57BL/6, to accommodate syngeneic tumor studies using cell lines derived from either strain background. One way to extend the utility of the JH mouse to more sophisticated syngeneic tumor experiments is to cross it to a mouse that is genetically humanized for a relevant target such as PD-1 or CTLA-4. Taconic can provide researchers with assistance in starting syngeneic studies with the JH mouse. Learn more at our website, taconic.com/JH.